Good morning, everyone. On behalf of NANO, I'm Dr. Balachandra, and I'm the faculty of botany. And today we are going to discuss about do a sort of a discussion on the NEET 2023 paper of botany. And first of all, on priority basis, remember most of the questions that come this particular year are focused onto the sort of a textbook. So it's basically conceptual base, and reasonably it has been moderate, barring a few questions wherein what happens is we have a little bit of analysis. So we straight on go along with relation to the sort of questions. Fine. Okay. Let's go to question number 1. So when you take 101, it states which micronutrient is required for splitting of water molecule during photosynthesis. If you all recollect, photosynthesis is basically a sort of an anabolic process, and photolysis of water is called as water splitting. It has been studied by Van Niel, and primarily during photolysis of water, what we understand is manganese is a sort of a micro element which is primarily involved with relation to photolysis of water. And so the right option is primarily 4, where manganese primarily helps in splitting up of the water molecule to liberate oxygen as well as the sort of a H plus. So water gets splitting of water with the liberation of H plus or the electrons and what is called oxygen requires primarily manganese as a sort of an element. So the option is primarily 4 with relation to question number 1, which is a direct question from the sort of a textbook. Sorry. Yeah. Now we go to question number 102. 102 is basically, it's from plant anatomy, from anatomy of plants. It says it's a statement question. You got to primarily ask about whether the statements are true or false. So when you look at the statement, statement 1 says endark and exarch are the terms often used in describing the position of the secondary xylem in the plant body. Remember, when you're talking in terms of secondary xylem, we are dealing with relation to secondary growth. And in secondary growth, there is no mention of endark and exarch. There's no mention of this. When you take statement number 2, it says exarch condition is the most common feature of root systems. So whenever we deal with relation to roots are always associated with what is called an exarch condition. So we understand that what happens is statement number 1 is wrong and statement number 2 is right. So statement number 1 is right. Statement number 1, so it says what happens is statement number 1 is incorrect, but statement number 2 is true. So the correct option is 3. Is primarily 3 here. So these again are, this is basic requirement, like you know what is a root and you know what is meant by secondary growth. A person who is primarily well read with relation to the basic concepts of plant anatomy can easily answer this question. Next, again, we go to the third question. Who provided the univocal proof for DNA as a genetic material was first proposed by? The options are you got Alfred Hershey and Martha Chase, Avery MacLeod and McCarthy, Wilkins and Franklin, and again Frederick Griffith. This has come primarily from the chapter of molecular basis of inheritance, and in molecular basis of inheritance we have DNA, the evidences, the first sort of a Nobel Prize evidences for what is called the DNA is a genetic material. If you recollect, Hershey-Chase experiment primarily deals with bacteriophages, and through bacteriophages, how does a sort of a genetic material get into the sort of a bacteria? And they had used radioisotopic nitrogen and phosphorus, and they tagged exactly whether the protein or the DNA gets into the sort of a bacteria. And their study of Hershey and Chase, for which they got a Nobel Prize, gives what is called the univocal proof. Now, what do you understand by unequivocal proof? When you say unequivocal proof is the final proof. The final proof, or which is very much evident in all organisms, DNA is a genetic material. That was primarily proved by Hershey and Chase. Prior to that, what happened is we had Avery, MacLeod and McCarthy. If you remember the experiment of Avery, MacLeod and McCarthy, they had primarily taken the transforming principle and they said that the transforming principle is the DNA. And it was based on primarily the experiment of Griffith. Griffith primarily said there is something called as a sort of a transforming principle which primarily killed mice. That was the first sort of a basic experiment on to establish whether DNA is a genetic material. They only knew that there was a transforming principle and they could not primarily convince whether it's the DNA, RNA or the protein. And later on we had Avery, MacLeod and McCarthy who had primarily used proteases to kill to remove proteins and RNases to remove RNA, and then they had also tried DNases to remove DNA, and they had established that DNA is the transforming principle. But the first universal proof or the uni-equivalent proof that DNA is a genetic material in all living organisms right from bacteria to elephants is primarily given by Hershey and Chase, and Hershey and Chase is a sort of their experiment of what is called the kitchen blender experiment using radioisotopes has been one of the sort of landmarks of molecular biology. Whereas when you take Wilkins and Franklin were again two important scientists who are primarily associated with the discovery of the DNA, and they had studied the sort of an X-ray diffraction studies. And remember, Wilkins and Franklin primarily they had established what is called the base equivalence of the Chargaff's rules, and that was the basis for how the sort of a DNA helical structure is formed. So when they ask you primarily questions of this sort, you got to primarily you got to follow the word. The key word to follow is unequivocal proof. It is the proof that DNA is the first genetic material. First boy and it was first proposed. So the option is A is 1. Now, when you go to question about 104, the phenomenon of pleiotrophism. Now, what do you understand by pleiotrophism? Pleiotrophism means one gene having multiple phenotypic effects. A single gene which expresses in different characters is termed as pleiotrophy. A single gene having multi-phenotypic expression is primarily called as pleiotrophism. Pleo means many, trophism means expression of a single sort of a gene. In the context of this, you're going to primarily understand what happens is, if we have maybe you inherit a sort of a character for intelligence. Intelligence could also lead to what is called musical ability, reading ability, and also memory ability. So this is what is called a sort of it's called a pleiotrophic gene. And a pleiotrophic gene normally is expressed even in plants. In plants, supposing there is a gene for pigmentation, the gene for pigmentation is also associated with what is called phytochromes. It is also affected with relation to trapping of the solar energy. So one gene, many effects is what is called as pleiotrophy. So the option here is 2. Remember, it is not 2 alleles for controlling a single trait. It's not 2 genes affecting a single character. It's not several alleles of a single gene controlling a single crossover. It is very clearly given as option number 2, a single gene affecting multiple phenotypic expression. Then we go to 105. When you go to 105, it says upon exposure to UV radiation, DNA stained with ethidium bromide will show which color? It will show either bright blue, yellow, orange or red in color. So if you take the chapter of molecular base of inheritance, primarily what happens is we have the extraction of the DNA. So when we do the DNA extraction on a gel column using a technique which is what is called as polyacrylamide gel electrophoresis, what happens is we are separating the sort of a DNA fragments. The DNA fragments come out primarily by gel separation. It comes out in the form of bands. So when you take the bands, when you are removing the bands, the process is called as elution. And after you cut the sort of a gel band, what happens is you look under you view it under UV light. So when you view it under UV light, what happens is you add a dye which is what is called as ethidium bromide, and the ethidium bromide under UV light gives what is called a sort of an orange band, and the orange band is primarily indicator of the nature of the DNA. How do you identify a DNA during gene recombination, or how do you identify that the part of the DNA is primarily cut by means of restriction enzymes? How do you identify a particular sort of a DNA is primarily loaded out from a sort of a phage is primarily by means of what is called bright orange coloration. That is option number 3, which primarily says DNA gets stained. So ethidium bromide is a sort of a stain under which gives in what is called orange coloration. So this is again all the questions till now have been direct evidences from your sort of a textbook. Now we go to 106, which is primarily from sexual reproduction in higher plants. To understand, large colorful fragrant flowers with nectar are primarily seen in bird, bat, wind or insect pollinated plants. Whenever we have insect pollinated plants, that is whenever we have entomophily, or whenever we are dealing with relation to insect pollinated plants, they're always the flowers are large for attraction. Remember, they primarily have pollination guides. They have pollination guides. That is, whenever you take a particular flower, you will have purple lines or white lines, which primarily are recognized by the compound eye of the insect. And again, it's very colorful. And again, it gives fragrance. And most importantly, nectar. And remember, nectar is primarily a sort of a reward. A pollination reward. A pollination reward is excess of presence of nectar as in the form of a food. So the correct answer here is 4, and this is the basic sort of a fundamental sort of a basic botany question. A basic botany question which says what are the characters of insect pollinated flowers. Now we go to the next one. Among eukaryotes, if we take higher plants, higher organisms, or what are called as eukaryotes, remember DNA replication, that is the doubling of the DNA making DNA. Doubling of the DNA always occurs in the S phase. S refers to the sort of a synthesis phase. So what is meant by synthesis? Synthesis means DNA duplication. DNA duplication means a DNA strand making a sort of a DNA strand. And this is a part of what is called the cell cycle. It is a part of the sort of a cell cycle, and S refers to the synthesis strand. So direct replication in the case of eukaryotes occurs during S phase. That is option number 1 is right down here. Now we go to 108. What do you understand by express sequence tags? Expressed sequence tags, that is ESTs. ESTs primarily refer to like all the sort of a genes, all genes which primarily code for what is called RNA. You know, primarily what happens is a gene is a stretch of DNA. And when you take a gene is basically split. A gene has got expression sequences which are called as exons, and it has got intervening sequences which are called as introns. So expression sequences code. So expression sequences code in a particular pattern. And what do you understand by a genetic code? A DNA makes an RNA and an RNA makes a protein. When a DNA makes an RNA, it is called as transcription. We are talking in terms of expression. We are dealing with relation to expression, and we are dealing with exons, which are expression sequences. And if the expression sequences, all the genes which code for RNA are switched on, they are called as expression sequence tags. So all genes which are on and primarily which code for the sort of a transcription, which are code for transcription, are primarily called as expression sequence tags. So the option here is basically 4. We have the 4th option. So again, every statement down here, if you check up on your sort of an NCERT book, most of them would be, you could be finding them on that particular sort of a page. Fine. Now, among the evil quarter, this is ecology questions. This time we have got a lot of questions from molecular biology and genetics as a sort of a unit, and also many questions have come from ecology. So, the focus of NEET 2023 had been like how many questions are basically coming from different sort of units. So, when you take the ecology unit is always on a priority basis. So, among the evil quadrant, which one is considered the most important cause driving extinction of species? What primarily promotes or primarily accelerates the extinction of a particular sort of a species? So, we have got over exploitation for economic gain, that is primarily because of primarily anthropogenic. And alien species invasion, supposing a foreign organism comes in, like if you got Parthenium coming into a sort of a forest, or you got Econia going in a sort of a water body, are always co-extinctions. That is, you lose a sort of a you lose a sort of a link in the sort of a food web or the sort of a food chain. Otherwise, we have habitat loss and fragmentation. Remember, habitat loss and fragmentation is the most important driving cause for extinction of species. So when you say habitat loss, like if you take a biosphere reserve, you take a sort of a national park, you take a biosphere reserve, and in a biosphere reserve, what happens is, you think you've got a Periyar wildlife sanctuary and you've got elephants going through. You're blocking the sort of a habitat, you're blocking the habitat loss, you're building a sort of a bridge, you're building a sort of an urban highway in the sort of a forest, whereby what happens is animals cannot cross. That is primarily called as habitat loss. So habitat loss is attributable to, say, some wildlife species like lions, tigers, like snow leopards. Snow leopards primarily from the Himalayas when they come down, or else primarily when there is only, like if you've got a one-horned rhinoceros, primarily in Kaziranga. So you remember what happens is, always when there's a break in the sort of a habitat and there's a fragmentation, that leads primarily to extinction of the individual species. So what happens is, we have to primarily bridge, like if you take a biosphere reserve, in a biosphere reserve, if the habitat is broken, again we are going to restore the sort of a habitat. So when we restore the sort of a habitat, what happens is, we build big sort of a bridges between habitat to habitat. So supposing this has been a sort of a biosphere reserve and there's a habitat fragmentation, and here we got the elephant corridor and elephant corridor, we again need to develop what is called a sort of an elephant corridor. An elephant corridor will enable the sort of a species to cross from one place to the other place. So habitat loss and fragmentation, which is basically due to development, due to unsustainable development, is the reason for the most important cause for extinction of species. So remember, the option is 4, and it's a very important sort of a thing on with relation to conservation. In the case of, it's a conservation issue with relation to ecology. Now let's go to other one. This is primarily from the chapter of respiration. So given below are two statements, one is labeled as A and the other is labeled as reason, and you're going to assert. Like assertion number A says ATP is used in 2 steps of glycolysis. Remember what happens is, we utilize ATP. Remember, ATP is called the energy currency of the cell, and ATP is utilized primarily. The reason is, the first ATP is used for converting glucose to glucose 6-phosphate, and the second ATP is used for the conversion of fructose 6-phosphate to fructose 1,6-diphosphate. Whenever ATP molecule is added to glucose, glucose is a basic substrate in cellular respiration, and glucose is a metabolic product. And whenever you add a sort of an ATP, it's called as priming. So the first 2 reactions are what are called as priming reactions, and priming reactions refers to charging reactions. And in charging reactions, what happens is, when you add adenosine triphosphate, the terminal phosphate, which contains 7.3 kilocalories of energy, that gets logged on onto the sort of a glucose molecule. So supposing we have glucose getting converted to glucose 6-phosphate, that means your phosphate molecule, the phosphate, gets added to the carbon number 6. And again, when you take the second ACP, second ADP, when you take fructose 6-phosphate, fructose 6 carbon is converted to fructose 1,6 carbon. That means again your sort of phosphate gets attached to what is called carbon number 1. So there is like there are two charges over here, primarily, which is because of ATP. So it says what happens is, in the light of the above statement, choose the right answer. It primarily says that A and R are true. We understand statement number 1 and statement number 2 both are true, and R is the right explanation of A. Primarily because ATP is the helping and conversion of glucose. So that is one of the reasons why it will get converted to glucose 6 and fructose 1,6 to fructose 6 to fructose 1,6-phosphate. So the option is 4. So reading the statements and analyzing the contents is very important in the context of biochemistry, and biochemistry and physiology play a central role with relation to any sort of a competitive examination. Now let's come to the other one, 111. What is the function of tassels in a corn cob? Whenever you take a sort of a maize, when you take corn is called as maize. Maize is termed as zea maize, and you understand what happens is, the male inflorescence is called tassel. The male inflorescence is called as tassel, and the female inflorescence is what is called as cob. And remember, if you take a maize plant, where you have a sort of a cob, the cob is present on the lowermost portion of the particular sort of a plant. So when you take the sort of a cob on the maca cob, what happens is there are a lot of styles. And this one entirely is so again on the male you got the male inflorescence, which is what is called the sort of a tassel. You got the tassel, and this is called the cob. So it says what are the functions of the tassels on a corn cob. When you take the cob, on that again you got silky hair-like structures. So what are the functions of the hair-like structures is to trap the sort of a pollens. Basically to trap the sort of a pollens. It is a method, it is an adaptation for wind pollinated crops. For wind pollinated crops, to trap most of the pollens, what happens is they have either a silky style or you got the sort of a tassels which primarily help in trapping of the pollen grains. The option here is 1. Option here is 1. Next, the historic convention of biological diversity, the Earth Summit, was held in Rio de Janeiro. Remember names of scientists, years of very important conferences. If you look at the previous papers, in the previous papers they have asked a lot of questions primarily in ecology with relation to years. So the Rio generation or the Earth Summit, when you take the Earth Summit, which is primarily occurred in Rio, was a sort of a game changer, and it primarily had occurred in 1992. So when you take the Rio Declaration is a very important declaration with relation to biodiversity, which says how do you conserve biological diversity, and it says primarily that humans are at the center of concerns. We say that the humans are the center of concerns in relation to biodiversity. So it has been a very important sort of event. 1992 has been 20 years after the discovery primarily, or after the starting of the environmental crisis. The environmental crisis primarily started in 1972, and after that we have got primarily 1992, 20 years after that we have the Earth Summit, and the Earth Summit of the Rio Declaration is very, very important, and it is primarily a sort of a move primarily to establish the importance of biological diversity on planet Earth as well as for conservation. So it's a very important marker sort of a date, primarily the Earth's, and so that's why it is called a historic convention. The historic convention is all people meet, and primarily they take big decisions with relation to biological diversity. So BD, biological diversity and conservation, are the driving forces in environmental movements and environmental issues world over. Next we come to identify the character. So even this one is primarily from ecology. So we have a lineup of lot of ecological questions. Remember, when you take this, you understand detritivores perform fragmentation. Detritivores feed on dead organic matter and they break down. So we have two important things. You got to understand decomposition and disintegration have been the very popular words which have been coming primarily in all India medical tests. So primarily when you take detritivores they feed on dead organic material and they perform fragmentation. That is very important. They break it into smaller pieces. And the humus is further, the organic component of soils, primarily the chemical component is called as colloids and the organic component is called as humus. Humus is rich in organic matter. It's further degraded by microbes during mineralization. So when you have humus, remember, humus has got organic matter and it is broken down by microbes. And what happens is minerals like nitrogen, phosphorus, sulfur, phosphorus, everything are primarily nitrogen, phosphorus, except sulfur, etc. are released. So microbes further degrade by mineralization, and water soluble inorganic nutrients which are water soluble go down into the soil and are precipitated by a process which is what is called leaching. So all along a soil profile, when you look at a sort of a soil profile, you got an A horizon, B horizon, C horizon and a D horizon. Remember, the horizons are of different colors. So why are they of different colors? Because they precipitate out different sort of an inorganic compounds which can primarily leach out and get colored because they get oxidized. So statement number A, B and C, that is option number 4, are right. Whereas when you take D, the detritus food chain begins with living organisms. It never begins with living organisms, it's only with dead organisms. And earthworms break down detritus into smaller particles by a process called catabolism. It's never been catabolism. Catabolism is a big biological process. Earthworms only pulverize it primarily, remember that. So the option is A, fourth option, which is A, B and C. Now let's come to a basic chapter called photosynthesis in higher plants. When you take photosynthesis in higher plants, remember the reaction center is in PS2 has an absorption maxima of, when you take PS2 and when you take PS1, remember PS1 is popularly called as P700, P680 is called as P680 nanometers is called as PS2, and we got two important photosystems. Now, what do you mean by a photosystem? A photosystem is a light harvesting complex, and the light harvesting complex is made up of chlorophyll and accessory molecules which are receptive to different wavelengths of light. And we understand photosynthesis occurs within the wavelength of 400 to 700 nanometers. PS1 refers to, it is called PS1 because it was first discovered the higher nanometers of light. The lower nanometer of light is what is called as P680, and it has got an absorption max of what is called 600 nanometers. So this is a direct question again with relation to primary photosystems and how are they designated in light reactions. Now, we come to what is called morphology, and remember in the case of morphology we got axial placentation. Axial placentation is common in the case of China rose, which belongs to Malvaceae, Petunia, which belongs to Solanaceae, and lemon, which belongs to Citraceae. Lemon belongs to primarily Rutaceae. So axial placentation is common, and these are standard straight statements from your sort of an NCERT book, which is primarily 115. The option is 3. Option is basically 3. And in tissue culture experiments, leaf mesophyll cells are put in a culture medium to form a callus. And what is the phenomenon called? So we have differentiated cells. We have differentiated parenchyma leaf mesophyll cells. Mesophyll cells are living. And what happens is you put them in tissue culture. And it forms, what does it go to form? It forms callus, an unorganized mass of tissue, which cannot know how to differentiate to form a sort of a root or a stem. An unorganized mass of tissue is called callus. That means the differentiated cells have lost the capacity to divide, and this is called as getting back to what is called the first level, which is what is called as de-differentiation. That is, it forgets about differentiation. Remember, differentiation, de-differentiation, and all are properties of what is called as totipotency. So a mature differentiated cell loses the property of differentiation and goes back one step back into what is called as de-differentiation. Then it goes to form a sort of a callus, and the process is called de-differentiation. Remember, de-differentiation, differentiation, development, and all are different themes, especially with relation to the sort of a chapter of plant growth and development, as well as what is called plant tissue culture, which comes in strategies of food production, strategies in food production. So here we have primarily the option is primarily 1. The option is primarily 1. Now let's go to the other one. During the purification process for recombinant DNA technology, we are talking about purification of the DNA, and we are looking at the recombinant DNA technology from primarily BT. And here what happens is addition of chilled ethanol. Whenever you add, remember what happens is when you take BT principles and processes. In BT principles and processes, you have the DNA, and we are talking about isolation of the DNA. Remember, we take a cell and we break it by means of cellulose or chitin, depending upon whether it's an algal cell or a fungal cell. And ultimately, we also break it again along with RNases to remove the RNAs, and we break it with proteases to remove the proteins. And ultimately, we get what is called the sort of a DNA. And the DNA basically precipitates. It forms a sort of a. It is just like a sort of a jaggery. Just like a sort of spool of jaggery. So what happens is it precipitates whenever you have chilled ethanol, and the chilled ethyl alcohol, when you add, primarily DNA gets precipitated. So this is again a sort of a direct question, and even you have a sort of a photograph of this particular thing in the NCERT book. So the removal of this particular DNA is what is called as elution. The term is elution, and we have got precipitation of the DNA using chilled ethanol. So option number 1 is the right sort of a thing over here. Now, when you go to 118, cellulose does not form blue color with iodine. Cellulose, normally what happens is whenever we have starch, what happens is it forms a sort of a color. And when you take cellulose, it does not form a blue color with iodine because it does not contain complex helices and it cannot hold the sort of an iodine molecule. Remember, it does not contain complex helices. So cellulose is a simple sort of a glucose polymer. And it's not complex, and thereby what happens is because it does not form a sort of a spiral, whenever you add iodine, the iodine cannot be held by means of the sort of hydrogen bonds. So here what happens is the option is 2, and again this is a straight question from biomolecules. Now, spraying of which of the following phytohormones on juvenile conifers helps in hastening the maturity process that leads to early seed formation? Remember what happens is we are good when you take gibberellic acid. When you take gibberellic acid is a phytohormone which primarily hastens maturity process. What do you mean by maturity process? When you say maturity process, that means it hastens malting, and especially malting during early seed development, and it is a direct question primarily from the sort of a textbook. So you got option number is 1, which is what is called as gibberellin. Remember, zeaxanthin is primarily related to maize. Abscisic acid, stress hormone. Indolectic acid is primarily helps in growth promoter, especially with relation to lens. Now let's go on this. When you take on pteridophytes, remember the pair of heterosporous pairs among these. Remember, among these the most common sort of thing is what is called option number 1, which is called Selaginella, is called club moss, and then you've got Salvinia. Salvinia is also called as, primarily it's called a sort of fern plant. So Selaginella and Salvinia are called as heterosporous pteridophytes. So remember, when you say heterosporous, we've got both microspores, we got microspores and megaspores, and heterosporous pteridophytes are very important because they form the basis for seed habit. The reason why the question has been asked is a student should primarily be knowing about what is the importance of heterosporous pteridophytes, and from pteridophytes have given rise to the seed habit. And you got Selaginella and Salvinia. So Salvinia primarily is also a very important sort of, it's a very important sort of an example. Next question, the process of appearance of recombinant nodules occur in which substage of prophase 1 of meiosis? When you take meiosis, when you take the substages, you got primarily leptotene, zygote, pachytene and diplotene. And remember, recombination nodules primarily occur in pachytene stage. In pachytene stage, you got recombination nodules. What do you mean by recombination nodule? When you take chromosomes, you find dot-like structures, big dot-like structures, and these are what are called as recombination nodules. That means primarily crossing over has occurred. So it is a final proof or the evidence for what is called crossing over. Recombination nodules occurs primarily in pachytene. Again, direct statements from your sort of a textbook. So which hormone promotes internodal petiole elongation in deep water rise? So when you take deep water rise, is primarily a sort of practice which is done in China. They go in boats and they harvest rice under the water. So when you take deep water rise, primarily what happens is you add what is termed as ethylene. And ethylene is which is a fruit ripening hormone. Here it is primarily used with relation to promoting internodal elongation or petiole elongation. So it helps primarily in petiole elongation, especially in deep water rise, ethylene. And again, this is again a straight statement coming from books. The frequency of recombination between gene pairs genetics on same chromosome as a measure of the distance between genes to map the position of chromosomes was first time used by the student of Thomas Morgan Hunt, whose name is Sturtevant. Remember this? Now, Sturtevant, we are talking about genetics. So in genetics, we have got primarily the mapping of them. We got gene mapping, that is what is the distance between the sort of a genes, and the gene map is primarily based on what is called as the distance between the genes is indicated by the frequency of recombination. What is the frequency in a population of 100? What is the frequency? And this has been studied by Sturtevant, and he had made what is called the gene map. And today the gene map is the basis for the human genome processing. So that is the reason why Sturtevant is used. So we got option number 2. So remember what happens is Sutton and Powery, chromosome theory of inheritance, Sturtevant gene mapping. Then you got Thomas Morgan Hunt, who's primarily has worked with what is called sex-linked and sex-limited characters with relation to Drosophila. So you got to understand names of scientists are true. And how many, this is from photosynthesis. Yeah, how many ADP and NADP molecules are required for the synthesis of one molecule of glucose during Kelvin cycle? So remember, Kelvin cycle is called dark reaction. It has been studied by Melvin Kelvin, who got a sort of a Nobel Prize on this. And remember, when you take glucose is always a sort of a 6-carbon atom. Remember, it is C6H12O6, and for every particular carbon, what happens is, for every particular carbon, you've got primarily 3 ADP molecules getting formed, and again, you got 2 NADP H2s getting formed, for one cycle. So for 6 cycles, it comes to 18 molecules and also 12. So the option is 1. So remember, the first option here primarily says for how many, for the synthesis of one molecule of glucose, how many turns we require is 6 turns. So approximately, so exactly what happens is you got this into, so you got 18 plus 12 are the ATP molecules and 12 NADPH molecules, which are primarily generated by the Kelvin cycle. So when you look at the sort of questions, as you understand halfway through, what happens is questions are not really honestly very tough, but they are very direct, and you've got to be well read and well informed about your sort of textbook and check on the sort of practice sessions. So here it says this is from a small sort of a thing which comes from bryophytes. Remember, in bryophytes, what happens is the first stage of the gametophyte is the life cycle of moss as a protonema stage. So it is correct, A is true. Protonema develops directly from the spores produced in the capsule. R is also true. Therefore, what happens is we have what is called option number 4 is the right sort of an answer with relation to moss, gametophyte, protonema, etc. Then we have RNA polymerase 3. Primarily, if you take molecular base of inheritance, RNA pol 3, we have got RNA pol 1, pol 2, and pol 3 is a very important enzyme, especially when you talk about eukaryotes. When you talk about eukaryotes, and they are responsible for the transcription of the tRNA. The way how you're going to remember is the transfer RNA or the soluble RNA is primarily transcribed by RNA pol, and it also transcribes 5S srRNA and SNP RNA, which is called as snRNA. So the option is 1. So this is again, I mean, when you're reading on molecular biology, you got to always understand what is the roles of RNA pol 1, RNA pol 2, and RNA pol 3. The thickness of the ozone layer in a column of air, environmental sciences, environmental issues. Thickness of ozone in a column of air in the atmosphere is measured by means of primarily Dobson units, DU. Option is 4. It's measured in Dobson units. Straight question again. And this is again from plant physiology. When you take, what do you call this? Transport in plants, yeah. The forces generated by transpiration can lift xylem to a column of 130 meters high, true. Transpiration cools the leaf surface sometimes to 10 to 15 degrees because of change of state, and evaporates cause also cooling. So what happens is we say that both the statements are correct, so the option is 4. Both the statements are basically correct. Transpiration cools the leaf surface, and again primarily the forces for transpiration can rise to 130 meters height. Again same statements are very easy. And when you take primarily photosynthesis, when you take ecology, in ecology, remember what happens is we got gross primary productivity and we got net primary productivity, and R refers to respiratory losses. So when you remove respiratory losses in a particular sort of a plant and you calculate, then you get what is called as net primary productivity. And remember, net primary productivity is calculated for tropical rainforests. It is calculated even for oceans. It's calculated for coral reefs. So that's a very important sort of thing for calculating biomass. What is the total yield in a particular sort of a crop? What is the biomass content of oceans, of tropical rainforests, is basically calculated here in terms of net primary productivity. And net primary productivity is what is the gross minus the respiratory losses. The standard sort of an equation which primarily comes in ecology. And when you take taxonomy here, this comes primarily from the chapter of morphology. When you take morphology, remember family Fabaceae differs from Solanaceae and Liliaceae. Name one very important diagnostic feature. How do you distinguish between Fabaceae, Solanaceae, and Liliaceae? Now look here, we are talking in terms of stamens, androecium stamens. And remember here what happens is, whenever you take Fabaceae, in Fabaceae what happens is you got 9 united plus 1 free. So this is called as diadelphous condition. Stamens are 9 united and 1, so they are in 2 bundles, 9 united, 1 free. And again, the anthers are primarily dithecous. This is monothecous, this is dithecous. So that is dithecous. So the option is basically 4. It's 4. Diadelphous and dithecous anthers are a characteristic feature of Fabaceae. In gene gun method, which is primarily used in BT principles and processes. Remember what happens is, whenever you're doing plant genetic engineering, when you're doing plant molecular biology, in plant molecular biology, you're taking a sort of an alien DNA or a foreign DNA, and you're introducing it into host cells, what you do is you bombard the sort of host cells or leaves by microparticles. On the microparticles, you put your sort of DNA on the microparticles, and the microparticles are basically made up of tungsten or gold. So you're bombarding the sort of host tissue primarily through a sort of a gene gun, wherein you're loading your sort of a DNA, and it gets through the tungsten and gold particles, and you use them as biolistics. They are what are called as biolistics. So this is a very common method of plant genetic engineering. So the option here is 2, remember. Okay. Moment of accumulation of ions across the membrane against the concentration gradient. So when ions primarily move against the concentration gradient, this is called as active transport. Remember that passive transport is you drop an ion, it has got a free fall. Facilitated diffusion, an ion is picked up by a small sort of a boat and it moves through the sort of plasma membrane, that is facilitated. Active transport is you're moving against the concentration gradient. It is just like supposing you have a person moving through a revolving door. You're going to rush through the sort of people and go against them. So similarly, ions move against a concentration gradient, and when they've got to move against a concentration gradient, it is just like you moving through a revolving door. You're going to put in a lot of energy. So there's a lot of there's a need for what is called ATP. So ATP dependent against the concentration gradient is basically called as active transport. So active transport is very, very important. The third option is very important. This comes under the field of membrane biology, and it also comes under primarily transport in plants. Which of the following stage of meiosis involves division of the centromere? Remember, the centromere is a sort of a central sort of a, it's a central space in a sort of in a chromosome. The division of the centromere divides and goes to form 2 daughter chromosomes. This is the characteristic feature of anaphase 2. Anaphase 2 is characterized by division of the centromere. This is a characteristic feature of anaphase 2 in meiosis, in the case of meiosis, and the option here is 2. It's a very important sort of an event which occurs during meiosis. Now, in angiosperms, what are haploid, diploid, and triploid structures in a fertilized embryo sac? Once you have a fertilized embryo sac as a consequence of double fertilization, remember what happens is you have primarily when you take synergids, synergids are helping cells. Synergids are always haploid. Then when you take zygote, zygote is always diploid. And when you take the primary endosperm nucleus, it is basically triploid. So the option here is option number 2. And how do you know about all these particular things? If you look at the sort of a diagrams and double fertilization in the chapter of sexual reproduction of higher plants, it's very much significant over there. And we understand which are haploid tissues, which are diploid tissues, and which are triploid tissues in a common mature embryo sac of angiosperms. Then we are going to 135. Okay, what does it say? It says primarily late wood have fewer xylary elements with narrow vessels. The wood which is formed xylem, the wood which is formed later on, primarily what happens is it is non-conductive wood, so therefore it has got narrow vessels. And cambium is less active in winter. In winter, what happens is there is presence of callose plugs. So what happens is a dividing tissue or a meristematic tissue like cambium is less active. So both the statements are true, and basically what happens is this is and R is an explanation of A. So option number 4 is right down here. This has come from plant anatomy during secondary growth. Okay. Now 136 comes from biomolecules. When you take biomolecules, remember when you take biomolecules, we are talking about enzymes, and you remember melanate inhibits the growth of a pathogenic bacteria by inhibiting the action of succinic dehydrogenase. The fourth option is right. This is an example of competitive inhibition. In competitive inhibition, you understand there could be 2 active sites. One could be basically blocked. So primarily melanate inhibits the growth of pathogenic by inhibiting what is called succinic dehydrogenase. So basic principles of biochemistry are there, fine. Now, let's come to this. This is a very important sort of a question. Remember, now how do you really go through this particular thing? Like, remember, iron primarily is iron. When you take iron is catalase. Then when you take zinc, that is B. Zinc primarily synthesis of amino acids, B1. So you got A3 and B1. Then you got C4. When you take boron, boron is primarily important for cell elongation differentiation. Remember, polon is also important for germination of the pollen grains. and then you got D, which is primarily molybdenum. So we have what is called option number 2 down here. So this requires a little bit of thinking. It comes from the chapter of mineral nutrition. It comes from the chapter of mineral nutrition. Now, which of the following statements is not correct? Remember, not correct is very important. Algal blooms cause excess organic matter and improve water quality and promotes fishing. No, it does not improve. Primarily, algal blooms primarily are toxic. Water hyacinth grows in eutrophic, leads to imbalance in ecosystems. True. Amount of toxic substance in industrial waste increases in different sort of, this called as biomagnification, and microorganisms in important organic matter in sewage causes death of aquatic organisms. So what is not true is option number 1. So you got to read, when you go to read the statements in a competitive examination, you got to read them very, very slow and think about them. So remember here, algal blooms causing excess of organic matter. Yeah, organic matter. And it does not improve the water quality, or it does not promote fisheries. That is a contradictory statement. So that is the false statement down in 138. So the right option is 1. And so that is a sort of a question which had primarily come with relation to basically environment sciences, as well as again with relation to biological classifications on algae. Now let's look at this. This is a very important, these are the sort of questions which are going to come in the future. This is a very good analysis question, wherein you say oxidative decarboxylation, glycolysis, oxidative phosphorylation, and tricarboxylic acid cycle. Remember, when you take tricarboxylic acid cycle, it is because of citrate synthetase. Let's go back. So you remember, you got options very quickly. You got to know how to catch the sort of options. And when you take primarily, when you take oxidative phosphorylation, is a characteristic of the electron transport system, oxidative phosphorylation. And when you take glycolysis, glycolysis is also called as EMP pathway. And when you take tricarboxylic acid, yeah, so we have got all the sort of options. Remember here, the right option is oxidative decarboxylation is pyruvate decarboxylase A2. Then glycolysis is primarily called as EMP pathway. So A2, B4. Then we got C1 oxidative phosphorylation. A2, yeah, B. How are we going? Wait a bit, wait 1 minute. We are going on oxidative carboxylation. That is a characteristic feature of pyruvate decarboxylase. That is, we got A2. Then when you take glycolysis, glycolysis is also called as EMP pathway. We got 4. So B is 4. Remember, A2, B4, C3 and D1. So the option here is 3. Primarily, that's the way how you go to match. So this sort of questions are primarily going to be the sort of a turnover questions for getting your sort of marks. Now, when you take Klinefelter syndrome, when you take genetics, primarily what happens in genetics, you understand that what happens is they have a masculine development and the individuals are sterile. These are the two important things. So the option is A and B. This is primarily what is called as class. So the option is 4. If you take human genetic disorders in genetics. And when you take this mutualism, both are benefited is mutualism, it is A4. And you got commensalism, one eats and the other is not benefited. So one eats and the other is not benefited. You got 1. Then you got amensalism and you got parasitism. In parasitism, what happens is one wins and the other loses. So it is C3. So it is D3. Yeah, D3. Yeah. So you have option of what is called 4 down here. So this is again very, very important. Primarily when you go with relation to mutualism, commensalism, amensalism and parasitism. Now here when you come to, this is from plant physiology. Remember, cohesion is mutual attraction of water molecules. Adhesion is primarily more attraction in what is called is attraction towards the polar surfaces. Primarily remember the plus and minus charges. And when you take surface tension, more attraction in the liquid phase. And when you take guttation is probably loss of in liquid water. So when you take primarily, how are the options down here? When you take cohesion is A2, yeah, A2. Then we have got adhesion. Adhesion is B4, A2, B4. So the option here is 4th option. We have the 4th option down here, which is just coming in. Fine. Now, what does this say? This again comes in ecology. It says Gauss exclusive, competitive exclusive principle. Supposing we got two birds sitting on a sort of a tree and competing for the same natural resource. That is what is called as competitive exclusion principle. So, in competitive exclusion principle, what does it say is that closely related species compete on the same resource cannot coexist indefinitely, and competitively inferior one is eliminated eventually. Statement true is this is true, and carnivores are more affected in competition than herbivores. This is a wrong statement. Always herbivores are more affected than competitors. So what we say is statement number one. We say that what is called, what does it say? It says statement number one is correct and statement number two is false. So we have got option number two down here. So we have a lot of questions coming primarily from ecology. Now this is from again from plant anatomy. So when you take plant anatomy, lenticels are lens-shaped openings. Bark formed in early is never called as hard bark. Bark is not a technical term, it's a non-technical term. Bark refers to periderm and secondary phloem, true. Pelagion is not a single cell, it's multi-cell in thickness. So what happens is we've got option A and primarily we've got option D. A and D are right. So what happens is we've got option 1 is right down here. So these are all direct statements from textbooks. And when it comes to primary gymnosperms, when you take gymnosperms in plant kingdom, remember pollen grains are released from microsporangium and carried by air currents. Yes, we have got the sulfur shower in the case of gymnosperms, and air currents carrying the pollen grains at the mouth of the archegonia, where the male gametes are discharged and pollen tube is not formed. This is a wrong statement because pollen tubes are always formed down here. So here what happens is A is true and R is false. So the option here is 2 with relation to primary gymnosperms. And here when you talk about recombinant DNA, remember what are the most important things. In recombinant DNA, what do you do primarily? You cut the sort of a DNA, you isolate the DNA fragment, you cut the DNA at fragments B, then later on you do isolation. Then after you isolate, what happens is you insert the recombinant DNA into the host cell. No, you do amplification, you do D, and then you have A. So the option here is 4. So which are all the different sort of the steps primarily with relation to recombination DNA is given down here. Then when you take here, what happens? What is the, you got to match the following. Remember when you take M phase, it's called equational division. M phase is called equational division. So all we are in between options number 1 and 2. And then when you take G2 phase, it's the interval between primary initiation and DNA replication. Remember G2 is B3. And we know quiescent phase is primarily inactive phase, quiescent phase. And G1 phase is one place wherein what happens is proteins are basically synthesized. So here what happens is we have B1. Sorry. So option here is 1 down here. Primarily in this. How many different proteins does a ribosome consist of? It is made up of 18. This is a direct question from your sort of a textbook. So when you take chemiosmosis requires primary membrane, protein pump, proton gradient and ADP synthase. This is from cellular respiration. Option is 4. And when you take the statements here, it says flower is defined as a modified shoot. This is a very important thing, and get condensed to form floral appendages. So both are right and R is the correct explanation. So option is 4. This is a basic sort of a concept in flower. In flower is a modified shoot and there's a condensation which is primarily seen in the case of lotus. So option is 4. Yeah, I think primarily what happens is we have gone through all the sort of questions which are primarily come in botany. So when you look at or when you are primarily analyzing the sort of a different sort of a questions here, all the questions are predominantly from the sort of an NCERT textbook, and it requires basically very intensive reading, because any student who's reading for NEET examination need to be passionate, you need to be very prudent, you require a lot of patience and you require a lot of perseverance, and a very good classroom environment wherein you learn. It should be more learner centered and practice of all the sort of questions on an MCQ and clarity of concepts is a sort of a key towards success in a sort of a NEET examination. So thank you all very much.